that brings us to mutations. So mute means to change. Okay, so a mutation is a change in the DNA sequence. So maybe it was supposed to be A, but instead it is A, C. Okay, so that is a change that was not supposed to happen. Okay, so this could be a mistake. One common misconception is that all mutations are bad. They are not necessarily bad, but you tend to hear more about the bad mutations. Okay, many people have mutations that doesn't affect them anyway. So for example, if you have one eye that's brown and one eye that's green, that's a mutation. That doesn't necessarily hurt you in any way. You just look different. So there's lots of mutations that you can have, and we just tend to hear more about the negative ones that cause diseases. But just know that mutations are not necessarily bad. So anyway, mutations in your sex cells, in your gametes, are not going to affect you, but they will affect your children, because the gamete goes to your children. Mutations in your body cells will affect you. So a mismatched nucleotide sequence can occur once per 100,000 base pairs. That causes replication to pause. So you have these enzymes that repair DNA. They proofread it and they repair the DNA. And so that reduces the errors to 1 for every billion base pairs. Okay, so then if you have any that survive the 1 per billion, then that contributes to a gene mutation. Don't forget, you also have those checkpoints. You got a G2 checkpoint that's going to check for mistakes when DNA was replicated. Okay, but if it gets past all those things, then you have a gene mutation. Sorry, it's bad weather. Thunder is getting really loud. So anyway, like I said before, if this happens to one of your sex cells, if this happens during meiosis, then it's going to affect the sperm or the egg. So then you don't see the effect, but your offspring, your children, will see the effect if that sperm or egg becomes fertilized, and becomes a baby. So anyway, occasionally you have random gene mutations that produce changes that make the individual better adapted to the environment. So you might have a helpful mutation. In fact, mutation is the only way to add a new trait in. Okay, so for example, humans have lots of different hair colors, but one hair color we do not have is blue hair naturally. So if there was such a mutation that caused humans to have blue hair, then that would add a new trait into the human gene pool. So this is really the only way to add a new trait. Sometimes those new traits might be helpful. It might help you adapt to the environment. So if you lived in a setting where blue hair was helpful, like if you were an animal, then that would probably help you. So think of it this way. An albino rabbit would not be very successful in Mississippi. It would probably get eaten by predators very quickly because it would stick out like a sore thumb. But an albino rabbit in an area covered with a lot of snow will probably survive better than a typical brown rabbit would. So different things. We're not just talking about humans here. Remember, when we're talking about these things, can help us to be better adapted to the environment. New traits like that can sometimes make us better adapted to an environment. Okay, so if it is a good mutation, if it is helpful or in helping you become better adapted, then these mutated genes will increase in frequency within a population. So that one rabbit will survive. That one albino rabbit, in the snow, it's going to survive, and then it's going to reproduce. And then you have a whole family of white rabbits, and then all that family is going to reproduce. And they were more successful than the brown. The brown ones, a lot of them died. And so you ended up with all these white rabbits. So you see an increase in the frequency or the number of times you see that white albino gene. And that's just one example. So a gene knockout is a process where a gene is non-functional. It's been made non-functional by a directed mutation. Okay, so a mutation occurs and it basically causes the gene not to work. Okay, it causes the gene not to be able to make a protein. Okay, so it doesn't get expressed. So a hairless mouse is an example of that. It basically knocks out the ability to express that gene for hair. This is Elizabeth Taylor. She is a very famous actress. She has passed away now, but Elizabeth Taylor is known for her violet eyes. Okay, she had a mutation which caused her eyes to appear violet or a purpley color. And so as you can see in this picture, they look very purple. She had a perfume named Violet Eyes. She was extremely famous actress. Again, her name is Elizabeth Taylor. This is a picture of her in her younger days. She died when she was 89, 90. She was pretty, on up there in age. And it hadn't been that long ago. It's been in my lifetime. She was a very famous actress, I guess, in the 60s, 70s, maybe, 60s probably, 50s, 60s. Anyway, so her mutation was a very beneficial mutation for her as an actress because she was known for her beautiful eye color. She also had a double row of eyelashes. Okay, so her eyelashes were so thick, she did not wear mascara, and she didn't have to. So she didn't have to wear mascara, so they could achieve like a very natural look with her because of her double row of eyelashes. Now, for some people, having a double row of eyelashes can cause like eye infections and problems, but for her, it really didn't. So this particular mutation for her gave her a unique eye color and a double row of eyelashes, which helps to explain one of the reasons why she would have been an extremely famous actress, other than her, just basic skill level. It would have given her kind of an advantage. Kind of like, an 8-foot tall person is going to be very easier for them to become a professional basketball player than someone who is 5'5". So anyway, here's some mutations. So if you'll take a look here, we got a couple different ones. So deletion mutation, that's this one up here. It's very simple. One piece of the DNA is deleted. Okay, so this would be the before, and then after the DNA is shorter. Okay, so a piece was deleted. Duplication, one piece of the DNA is doubled. Okay, so you'll see that particular sequence twice, so it's duplicated. Okay, so those two are very, very simple. Then you have some that, then you have insertion. Insertion's still pretty simple, where you insert a new piece, okay? So you insert a new piece of DNA. Inversion, okay? Inversion sometimes people struggle with. Okay, with inversion, they are flipped. Okay, like in math, when you take the reciprocal of something, so if your fraction is 1/2, to take the inverse, it would be 2/1, right? So you would flip it. So the same thing is going to happen. If the DNA is A, B, C, D, then it would be A, C, B, D. That would be an inverse. You would flip the B and the C. Then you have translocation. Translocation, it's going to change locations to a different chromosome. Okay, so basically, two chromosomes are going to swap. Okay, they're going to swap pieces of that chromosome. So if you look here, you got some of each one. That is different from insertion, because with insertion, it just inserts a new piece. They didn't actually swap. Okay, so coming over to the side here, frame shift. Okay, so on the left, we just talked about chromosomal mutations, which usually will affect many genes. DNA mutations are going to affect usually one or more particular DNA sequences. So you might have a frame shift mutation. So if you look, this is the top. Oops, sorry. So this would be my original code, and that will be the amino acid sequence. Okay, so if it shifts, and we don't use this C, okay, and that causes a shift, it causes us to use ATT, CAC, ACG. Notice that a lot of your amino acids are changing, so it affects everything after the shift with the frame shift mutation in DNA. Okay, so you have gene mutations. So a gene is going to change one or more DNA nucleotides. A lot of the times it's deleted or substituted. This can also be called a point mutation. So it's one particular point that's mutated. Okay, so if you add a new letter or delete a letter, so if you're supposed to have 20 letters and you add one in, now you have 21. Or if you take one away, now you have 19. That causes a shift, okay, when you're reading the genetic code. So that's called a frame shift mutation. So that means everything after that mutation will be messed up. So thinking about like getting out of sequence when you're marking answers on a Scantron. Okay, so anyway, a point mutation may change just one or a couple of nucleotides in a gene or even on a chromosome. Okay, so sickle cell anemia and cystic fibrosis are two genetic diseases that are actually point mutations. So even though it's one little piece that's mutated, it can cause a major disease. Like I said, sometimes you have these random gene mutations that are better for the organism. Okay, and in that case, if it's better it makes it better adapted to the environment, that will make you see it more often. So again, a gene mutation is where one gene is changed. So this can happen when one nucleotide is substituted for another, or if you add or take one away. So this can cause a protein to change so much that it cannot work right. So here's a great example of frame shift mutations. Now, keep in mind that with these mutations, your DNA is going to be A, T, C, and G. Okay, so I'm using this sentence to show you how big of an effect it can have. Okay, so your example is up top. So this is your original. Okay, so what it's supposed to say is, the rat hid and the cat sat and got fat. Notice these are all 3-letter words, like your mRNA codons. Okay, so in a substitution mutation, you would have the pat, so we substituted this P here. The pat hid and the cat sat and got fat. Okay, so if you notice, this is the only amino acid that would be changed. So this would not be a frame shift. This would just be a point mutation. All of these are point mutations, but it may or may not be frame shift. Okay, now if you look at insertion, the rat hicks and blah, blah. Okay, so here we inserted the X. So you still have the word hid, and the cat sat and got fat. So you still have all the words, you just inserted an extra letter. Now because of that, every one of these amino acids will now be altered. Okay, so that shifts it all down and it messes it all up. Same thing here. We deleted the T there. Okay, so hid and the cat sat and got fat. So because we deleted that one letter, every single one of those amino acids after that point will be affected. And that is the shift that we were talking about. Okay, so again, this is the same picture. Did not mean to duplicate that. Sorry about that. Okay, that brings us to karyotypes. So karyotypes can show a change in the chromosome number. Okay, so a karyotype is basically a photograph or a picture of a person's chromosomes. And we've talked about karyotypes before. And so they're great for diagnosing Down syndrome. That's one of the most common diseases diagnosed with a karyotype. So your homologous chromosomes are going to be paired up, and they're going to have the same. So chromosome pair number 1, it's going to have genes. Each of those chromosomes is going to have gene for a particular trait. Okay, so your number of chromosomes can be studied using a karyotype because it's simply a picture of your chromosomes. Okay, so here we have 2 karyotypes. Okay, so if you look here, notice there are 2. Okay, so one would have came from your mom, one would have came from your dad. And notice that your chromosomes are arranged from largest to smallest. Then you have your sex chromosomes as your 23rd pair. Okay, so obviously this is a female and this is a male. And so I can look at this karyotype and see, okay, I have 2 for every chromosome. Okay, and so I can know that this would be a normal female and a normal male karyotype. Okay, so if you take a look at this karyotype, they've highlighted that it has 3 instead of 2 chromosomes on number 21. And so trisomy 21 is Down syndrome. Tri is 3 and somy is chromosome. So it has 3 chromosomes on number 21. So anyway, each of your individual 46 chromosomes has thousands of genes on it. All of those genes play different roles in determining how you look, how you develop, and how your body functions, if your body functions correctly. You have to have all 46 of those chromosomes. Most of the time, if you are missing one chromosome, or if you have one too many, then the individual does not survive, and often it would be a miscarriage. So I've talked to y'all a little bit about that before, how my husband and I had miscarriages, and we, after our second miscarriage, we had our DNA karyotype to double check we didn't have a chromosomal abnormality, and we did not, fortunately. But anyway, so if you're missing one chromosome, it's very likely that you wouldn't survive to become a baby. If you did, you would have some severe problems. But even if you have an extra, there's not very many cases or diseases where you can have an extra or missing a chromosome that is able to survive. But if it's a specific chromosome, then it is possible. For example, Down syndrome is trisomy 21. That's one of the chromosomes that you can survive with if you have an extra. So a karyotype, again, is helpful for diagnosing Down syndrome. Down syndrome individuals will have 47 chromosomes because instead of 2 on number 21, they will have 3. Okay, so a karyotype can tell you if you have an extra copy or if you have a missing copy. Okay, so it cannot show individual genes, though. So, how I would look at a karyotype, or how I would get a karyotype. So they would take body cells, so that you can see all 46 chromosomes. So they would take some body cells, and they would look at the nuclei, and usually it's going to be prophase, probably metaphase, that they would look at in mitosis, because remember, you can't see DNA when it's in interphase. So they stain the chromosomes, and then they photograph them. And then they arrange those homologous pairs, and if you notice, they were arranged by size and shape, and anyway, then they look at them and analyze it. So for us in particular, they took our blood and they looked at our white blood cells, the nuclei of those. Anyway, so all they did was draw one tube of blood, and that was that. I actually still have the microscope slides, and I have my karyotype for me and my husband as well. So anyway, the karyotype analysis is going to reveal any major or gross chromosomal abnormalities. So, how you would get Down syndrome, or a different number other than 46 number of chromosomes. Non-disjunction is how. So non-disjunction is a kind of different type. It's a chromosomal mutation. So what's going to happen is the chromosome pairs do not separate during specifically meiosis. Okay, so what happens is normally in metaphase 1, your chromosomes are lined up in 2 rows, right? And anaphase 1 is supposed to split the chromosomes, and they're supposed to go to opposite sides of the cell. Well, if anaphase does not do this properly, then what will happen is you'll end up with your 4 cells produced at the end of meiosis, and one of them will be missing a chromosome. So that will be a monosomy. Then one of them will have an extra. That will be a trisomy. Okay? If one of these is fertilized, then that becomes a child with, a trisomy. If this one becomes fertilized, then that becomes a child with a monosomy. So anyway, Down syndrome is caused by the extra 21st chromosome, so it's because of this non-disjunction mutation. So that's the most common one. Another one is cat-eye syndrome, which is also a trisomy. It's just not number 21. It's a different chromosome. Cri-du-chat, which is a monosomy, and then Turner syndrome, which is one of the sex chromosomes rather than one of the autosomes. So here is a more digitized version of a karyotype. Okay, so if you notice here, we have an extra on number 21, and it is a male. Okay, so I can see their gender on a karyotype, and I can also count to make sure that all the chromosomes are in pairs instead of having one less or one too many. Okay, so here is what individuals with Down syndrome look like, and just a few characteristics that you might see. So my uncle actually has Down syndrome. He is 51 years old, so he has lived a very long time for someone with Down syndrome, and he actually is considered a dwarf. He is about 4'3", I think. He's just adorable and the sweetest thing you'll ever meet. But anyway, so he does have Down syndrome. Other chromosomal mutations. So again, it's very similar to gene mutations. Genes are pieces of chromosomes. So anyway, again, a mutation is a change in the organism's structure, specifically of DNA. Sometimes chromosomes can break off and cause different mutations. So first off, a deletion mutation. Okay, so you would break off a piece of a chromosome. So if you lose a whole leg of a chromosome, that would be a deletion mutation. Okay, so they're going to lack genes. So usually this is fatal to the zygote, so it would not actually ever develop, and it would become a miscarriage if you even knew you were pregnant. The next one is duplication. So this is where a chromosome fragment attaches to the homologous chromosome, and then instead of one copy, it has 2 copies. So it has more copies of the genes than is needed. So here you have a picture of a deletion mutation. Okay, so we have before and after. So if you notice, they just deleted part of it. And then in duplication, you had 1, and now you have that 1 in 2 spots. So it's been doubled. Okay, then you have inverse mutation. So again, like I said, they flipped the letters there. So it's where it is flipped, but it's reattached, but in the reverse position. And then translocation is going to break off and reattach to a different chromosome. So it's going to be in the wrong place. So I really like these pictures with the letters because you can see how it should be A, B, C, D, E, F, like down here. You can see how it should be A, B, C, D, E, F, G, and the other one should be H, I, J, K, L, and you can see how they attach to the wrong spot. Why you get mutations. So mutations mutagens increase the risk or how often you see mutations. If you're around a mutagen, that doesn't necessarily mean you're going to get a mutation, but it just increases the likelihood of you getting one. A carcinogen is a mutagen that causes cancer. Okay, so UV radiation from the sun, of course, can cause skin cancer. Tanning beds, of course, same thing. X-rays, tobacco, of course, we know can cause cancer. asbestos, benzene, just different chemicals. So things that we know can cause cancer. An oncogene we've referred to several times now. So an oncogene is a cancer-causing gene. Okay, so I've referred to the BRCA1 and BRCA2 genes that cause breast cancer. Now, most cancers are not caused by oncogenes. I'll just say that at first. But there are some cancers that are caused by oncogenes. Like I said, there's not very many of them. But, so oncogene is just what it sounds like. Onco means cancer. So it's a gene that causes cancer. Okay, so what happens is this is a gene that had a mutation that caused a malignant change in the cell. So that change was harmful to the cell, but it didn't kill the cell. Okay, so what happens is a lot of the times these cancer cells have more than one nuclei, so they're multinucleated, and they are a weird shape, so they're elongated. So that's why if you see like tumors, the cells might look real weird looking rather than like smooth and how it's supposed to look. So that says many types of cancer and warts. So Klinefelter syndrome is just one thing that I wanted to talk to y'all about as far as mutations go. We will get into a lot more genetic conditions when we get into the next unit, which is genetics, which will be after exams. Anyway, so Klinefelter's is just one genetic condition that we'll talk about that's a result of a mutation. So technically, someone with Klinefelter's is going to have, instead of XX or XY, they're going to have XX, which would be female, Y, which would create male characteristics. So XXY. So they have an extra X chromosome. So they would have 47 chromosomes. So they would have 2 X's and a Y. So they're considered male because they have a Y chromosome. And so they believe this is 1 in every 500 males or 1 in every 1,000 births. And of course that's going to, vary a little bit. But anyway, people with Klinefelter syndrome generally are taller than average. Their arms and legs are like longer than average, and they are usually sterile. So that means they're not fertile, that they basically can't have kids, or they would struggle with having kids. They would struggle making eggs or sperm. Anyway, a lot of the times they will have some amount of mental retardation or some mental type struggles. And so if you look here at this karyotype, you can see there's 2 X's like you would have in a female, but then you also have a Y present. So here is a couple of instances where you would see, so you have your XX, which is normal female, XY, which is a normal male, but then you have a condition with just 1 X, and then you got your XXY, and there's a couple others. So anyway, this is just showing you some chromosomal abnormalities or mutations. So if you end up with an extra chromosome like this, that is going to be the result of non-disjunction during meiosis, usually. So this just shows a person with Klinefelter's kind of symptoms. Again, they'll be considered male, but they'll have poor beard growth, and that can kind of vary. Boys struggle with beards anyway until they reach a certain age, but also it depends on your genetics. Like if you have American Indian in your genetics, they tend to not have a lot of body hair, so they might not grow a good beard. Anyway, so men typically have wide shoulders, but someone with Klinefelter's is going to have more narrow shoulders like a female and wider hips like a female. Again, they have XX, so they might have some female characteristics like the narrow shoulders, the wide hips, the breast development. And then they have the Y, so they might still have testicles, but they'll be smaller in size. So just, it's almost like male and female characteristics kind of battling. So anyway, like I said, we'll talk about more genetic conditions. in the next unit. And so this was just kind of an introduction of 1. So if you will, don't forget to watch the next video about genetic technology, so DNA technology. So that'll be next.